Okay, welcome back. This week we're looking at what the textbook is calling Chapter 5, so ignore that. This is actually Chapter 5, the reproductive system. They kind of divide the reproductive system into two chapters, the textbook does. The first one, Chapter 5, talks about the actual systems, the anatomies, physiology of the reproductive system. And then Chapter 6 kind of gets into what happens after fertilization. So it gets into the development and whatnot. So we'll do two chapters on reproduction. And let's see what we got here. So there are two genders, obviously, and for the males have it a little bit easier. Their job is to contribute the sperm, produce the sperm, store the sperm, and then deliver the sperm cells. Females, their job is to produce the egg cells, store the egg cells. And then they actually also provide the site for fertilization. So where the sperm and egg meet, that's going to be happening inside the female reproductive structures. Populations that reproduce using sexual reproduction, what they do is they produce individuals using a variety of genes. So we have individuals that are being produced with genes that are coming from two different sources. So when this happens, the result are going to be offspring that show greater genetic variation because they're getting their genes, their DNA from two different sources. All right. So what does this do? Well, this enhances the population. It makes the population stronger. When there's variations in population, the individuals are different from each other. And why is that a valuable thing? Imagine if we were all the same, if we were clones of one another, like some bacteria are. And then a virus showed up that was able to kill some of us. Well, if we were all clones of each other, that virus would then be able to kill all of us and probably will. But we're not all the same. We are genetically different. We have variations. We are the products of sexual reproduction. And the variations that exist within our population allow us to better adapt to the environment, especially when the environment changes. And having a virus come into the environment is a good example of the environment changing. Okay, so this leads to populations that are fitter, more fit. So there's evolutionary significance to being able to reproduce sexually. All right, this word gametes over here, forming gametes. There's two types of gametes. There's male gametes and there's female gametes. The male gametes are sperm cells, and the female gametes are egg cells or oocytes. And they are produced by a special type of cell division. It's called meiosis. And what's unique about gametes is the fact that they have a haploid number of chromosomes. So what does that mean? That means all the cells in your body have 46 chromosomes except for the gametes. The gametes have half the number of chromosomes. Half the number. I don't know why I wrote that. It says it right here. So why is it important to produce cells that have half the number of chromosomes that we're going to use for reproduction? Because when you take a cell with half, a sperm cell, and you combine it with another cell that's also half, well, then you have a whole, right? Then you take a sperm cell that has 23 chromosomes, and you combine it with an egg cell that has 23 chromosomes, then you're going to have a zygote that has the correct number of chromosomes, 46 chromosomes, which is the number that we have in all of our cells. All right, so we take a haploid sperm cell, a haploid egg cell, and we create a diploid zygote. Now, all the cells in your body are diploid, meaning that there's two sets of chromosomes. If we use the letter N to symbolize the number of sets of chromosomes, your sperm and egg cells will have one set each, right? But when we combine them, the diploid cells will have two sets, one set from mom and one set from dad. So a lot of times you'll see that written as 2N, as N equals sets of chromosomes. So forming gametes is very important, right? But it's not the only function in the reproductive system. There's other functions, bringing on puberty, maintaining reproductive abilities, bringing on secondary sex characteristics. These are all mostly controlled by hormones. If we look in both the male and female, we're going to see gonads. So the gonads are the structures that produce the gametes. All right, so we're talking about testes and ovaries. It's the testes that produce the sperm cells, and it's the ovaries that produce the egg cells. There's ducts involved, there's glands involved, and there's other supporting structures involved, and we'll look at all of those. A lot of times you see this word, this root word in science and biology, genesis. This means the creation of. Even in the Bible, it means the creation of. That's the first chapter in the Old Testament, right? And of course, gametes are sperm and egg. So this term gametogenesis refers to making or creating sperm cells and egg cells. In males, the specific type of gametogenesis is called spermatogenesis. So this is creating sperm cells. All right. So this happens as a result of a special form of cell reproduction that called meiosis. Remember, meiosis is that type of cell reproduction that produces haploid cells. And we start with one parent cell with 46 chromosomes, and the cells divide once. But we make copies of each of these, and then the copies split, and we're left with 4. I'm going to go to the next slide. 4 sperm cells that are haploid. So 4 haploid sperm cells from one parent, one diploid parent cell. And again, males are producing many every day, about 300 million. Oogenesis is what's going on in females. And again, this is the same thing. It's happening by meiosis. We're taking one parent cell and producing one large egg, and then 3 non-viable leftover cells that are called polar bodies. But again, we're taking one parent diploid cell and producing a haploid egg. These 3 are going to be discarded. They're not going to be usable because the egg is taking most of the cytoplasm. All right, so the egg has the potential to be fertilized by one sperm. The polar bodies are not viable and they are quickly degraded. All right, so all the eggs in the female are produced as they're developing, right? As the bodies of the females are developing in the uterus embryonically. One of the things that is also being produced are eggs. So by the time females are born, they have all the eggs that they'll ever need. All right, so how do we divide all the cells in the body? Think about skin cells. Think about growing. Well, that's mitosis. if we have 46 chromosomes in our cells, but let's say we had 4 in all of our cells. By mitosis, we would take one parent cell, diploid parent cell, with 2 sets of chromosomes. Here's one set. Here's another set. All right. And what do? We make copies of all the chromosomes, and we're left. The cell will split in half, and the originals will stay, and the copies will go another way, and then we have identical cells. This is mitosis is for growth, repair, that kind of stuff. Meiosis is a completely different story. We're starting with one diploid parent cell. These chromosomes make copies of each other, and then they divide. So we're left with haploid cells here, and then they divide again. And we're left with, if we're talking about males, it's 1, 2, 3, 4 cells from one parent cell. If we're talking about females, because the egg cell has to have a lot of cytoplasm, it steals cytoplasm from these 3, so it's much larger. So these 3 become discarded. They're not viable. We have one egg cell, but you see it has half the number of chromosomes than what we started. So this is meiosis. This type of cell division is only for producing gametes. That's it. That's the only time we see it. All the other cell division in the body that's happening is mitosis. All right, so let's start with the male. Like I said, the males have it pretty easy. You make sperm, you store sperm, you deliver the sperm cells. It's pretty easy. We could kind of consider the male reproductive system as just one long tube. At one end of the tube, we have the testes. This would be like the beginning of the tube, and it's in the testes that the sperm cells are made. And then from there, the sperm cells move along the tube and mature as they're doing so until they are released out the other end of the tube. There are other glands involved, like the prostate gland, the seminal vesicles, the bulbourethral glands, and we'll look at those. And what are they doing? They're adding things to the sperm to help carry the sperm through the tubes, to help protect the sperm as it's leaving the body, nourishing the sperm. Again, the sperm cells, they're going to be moving, right? And because they're moving, they're going to be using a lot of energy. So we have to provide nourishment so they can use energy. So this is a sagittal plane. So you could see if we were able to cut this open at this plane and look inside, what would we see? Well, if we wanted to highlight the structures of the reproductive system, it would start here. This is the testes, so it's in here that the sperm cells are made. After the sperm cells are made, they kind of hang out in this structure here and mature. This is called the epididymis. And then when the sperm cells are to be released, they travel through this tube called the ductus deferens or the vas deferens. This tube goes around the bladder. It goes through the seminal vesicles and then through the prostate gland, and then it'll meet up with the urethra. Now, if you notice, the urethra is connected not only to the vas deferens and is the delivery tube for gametes, for semen, but it's also the delivery tube inside the urinary bladder. Urine would be stored in here, and the urine would be leaving, also hooking up with the urethra. So this is a delivery tube not only for semen and sperm cells, but also for urine, all right, until it goes all the way through the penis and then leaves, all right? This is the scrotal sac. The scrotal sac allows the testes to be kept not so much close to the core in here where it's warm, but more housing the testes in a more external position or area where it's a little bit cooler, and the cooler temperatures helps with the division of cells. It helps with meiosis or gametogenesis. If we were to take one of these tubes, now inside the testes, there's millions of tubes, and they go back and forth and back and forth. And it's inside these millions of tubes that these tubes are called the seminiferous tubules. It's inside these tubes that the sperm cells are being made. If we were to take one of these tubes, pull it out and cut it and look inside, what would we see? Well, we would see the wall of the tube, right? And then we would see along the edges, we would see these diploid cells. So cells that have both sets of chromosomes, cells that are going to be the parent cells for meiosis. They're called spermatogonia cells. Again, 2N, that means they're diploid. As these cells along the wall divide, they go inward towards the middle of the tube. So from primary spermatocytes that are diploid, as they divide into secondary haploid cells that would be up in here. And as these divide even further, they divide into spermatids. And you can see the difference between spermatogonia and spermatocytes. Spermatids actually will show the existence of these tails. All right, so spermatids are immature sperm cells. So as these cells are turning into sperm cells, they're finding themselves inside the middle of the tube. And, we refer to the middle of any tube as being, we call this a lumen. This is the lumen. So the parent cells are away from the lumen, more towards the walls of the seminiferous tubules. But as they divide, those cells end up closer and closer to the lumen until we're left with spermatids themselves. And then they will swim through the tube, eventually until they get to the epididymis. Now, not all of the cells inside the testes are reproducing by meiosis and forming sperm cells. There are other cells outside the tubes. Here's a neighboring tube, right? This looks like the wall of a neighboring tube here. But there are cells that are in between the tubes. And these cells that are like over here, outside of the tubes, these are called Leydig cells. They're also called interstitial cells, and they're playing an important part in the formation of sperm cells because they produce testosterone. Leydig cells, interstitial cells. They produce testosterone, which is the hormone that is responsible for promoting the production of sperm cells. All right, so here we see Leydig cells up here producing testosterone. When the hormone testosterone is being produced, it's communicating with these cells. It's telling these cells, all right, come on, let's get going. Let's start dividing. All right, so the diploid spermatogonia cells will divide into spermatocytes, which will divide again into secondary spermatocytes until we have spermatids. All right, so we could see now we're in the lumen. The only types of cells that should be in the lumen are these immature sperm cells. All right. So it's at puberty that the spermatogenetic cells in the testes start to begin to produce sperm. So these divide into spermatogonia, which would be diploid, which divide into primary spermatocytes, which are also diploid. But when they divide again, they don't. They separate the copies of chromosomes. So each of the spermatocytes would be haploid, and then they divide again until we're left with 4 spermatids, each haploid. As they're dividing, they're pushing away from the wall and toward more and more towards the lumen. When they're in the lumen, they're going to be sperm cells. So we're starting with cells, spermatogonia cells that are 46 chromosomes. And what are we ending with? Well, we're ending with immature sperm cells that have half that number. So these are haploid cells. We can't produce diploid sperm cells because if you put a diploid sperm cell together with another diploid sperm cell, the result is going to be a zygote that has double the normal number of chromosomes. So we can't have that. We have to produce cells that have half, right? If there was, just think of like imagine it like you're combining pizzas, right? Imagine that we have a pepperoni pizza and then we have an onion pizza. So the pepperoni pizza, imagine, is the dad and the onion pizza is the mom, and they're going to have a baby pizza. Well, each one would have to contribute cells for half a pizza, right? So dad would contribute a cell that would be the equivalent of half of pepperoni pizza. And mom would contribute a cell that would be the equivalent of half an onion pizza. And when you put them together, what are we going to get? Well, we could get half pepperoni, half onion. Or we could get a whole pizza that has pepperoni and onion. Or we could get any different variety of any of those things. So I just want you to visualize that when we're talking about gametogenesis and creating sperm cells and creating egg cells, we're giving half of the needed amount of genes to create a person, right? So half plus a half creates a whole. So it's a very unique type of cell division, meiosis is. All right, so at the end of meiosis 2, the second division, we have these 2 haploid cells forming into 4 haploid sperm cells. So again, one parent cell produces 4 spermatids. There are other cells besides Leydig cells, which are remember, we talked about Leydig cells being also known as interstitial cells, producing testosterone, stimulating the whole process. Right? There's also Sertoli cells, and they're providing nourishment for the sperm cells as they are developing. If we were to look at the anatomy of the sperm cell, we have a head, and it's in that head that we have the DNA or the genes, the genetic material, in something that's called a pronuclei. There is on the head, there is an acrosome, like a little cap. And inside this acrosome, we have digestive enzymes packed in here. So what this does is once the sperm cell reaches the egg cell, there's barriers surrounding the egg cell, protecting the egg cell that the sperm cells actually have to burn a little tunnel through. So they would release these digestive enzymes that help them clear a path through those barriers. There's a protein barrier that surrounds the egg cell. and it's called the zona pellucida. So they would have to penetrate that in order to reach the egg cell. And they would penetrate that protein layer by releasing the digestive enzymes of the acrosome. There is also a midpiece. In the midpiece, there would be many, many mitochondria. Remember, mitochondria are the organelles of the cell that produce energy. And this cell is certainly going to be using a lot of energy because in order to move, it has to use its whip-like tail, right? And that requires a lot of energy. This whip-like tail is called the flagellum. So we would see a lot of mitochondria packed inside of here in the mid piece. A lot of mitochondria. This tail would be much longer than this. This is not drawn to scale. This tail would be much longer. All right, so the head, the acrosome, the midpiece, mitochondria, and a long tail. See, even the notes cut the tail, saying this is not the size. There'd be much more in between there. So there are some hormones that are responsible for controlling sperm cell production. In the hypothalamus is in your brain, right? It's releasing a hormone, and it's called gonadotropin releasing hormone. And that's going to trigger the front side of the pituitary gland to release two hormones. One of them is called luteinizing hormone. The other one's called follicle stimulating hormone. So what luteinizing hormone does is once it enters it from the pituitary gland into the bloodstream, it will travel down to the testes and communicate with those Leydig cells or interstitial cells to start producing testosterone. Remember, testosterone is the hormone that gets the cells, the spermatocytes, the spermatogonia to start dividing and producing sperm cells. So it's really triggering testosterone is the production of sperm cells. Okay, but these cells, again, like I showed you in the picture before, they're not inside the seminiferous tubules. They're right outside. They're right between neighboring seminiferous tubules. Follicle stimulating hormone stimulates Sertoli cells. Sertoli cells are going to be producing a protein called ABP, androgen binding protein. And what this does, this protein clings to testosterone. And the whole idea of clinging to testosterone is once the testosterone is being produced, we don't want the testosterone to leave the area. We need the testosterone to stay, right? So the tendency for any hormone once it's produced is to get picked up by the blood and travel. But we're trying to keep the testosterone localized. And that's what this androgen binding protein does. It keeps it near the seminiferous tubules and allows that hormone, testosterone, to stimulate the process of producing cells, and then which in turn should increase the sperm count. As the sperm count rises to an acceptable level, the Sertoli cells will then produce inhibin. Inhibin will stop the production of follicle stimulating hormone. All right. So we have what's called a negative feedback mechanism here. It begins in the hypothalamus, producing gonadotropin releasing hormone. That's going to stimulate the front. This is the front side. This is the back side of the pituitary gland. The front side of the pituitary gland to release two hormones. Luteinizing hormone. That's going to travel in the blood down to the testes, and it's going to cause the interstitial cells to produce testosterone. At the same time, follicle stimulating hormone is traveling down to the testes stimulating the Sertoli cells to produce ABP. ABP is going to make sure testosterone is staying localized until sperm cell numbers increase. Once sperm cell numbers are high enough, then the Sertoli cells will produce inhibin. And then inhibin is going to decrease the amount of follicle stimulating hormone, which should stop the whole process. So again, this is a negative feedback mechanism. Just one other example of maintaining homeostasis. Okay, so the sperm cells are being produced. Now they have to move through the ducts, through the lumen of these ducts, and until it's out of the body. All right, so the Sertoli cells inside the seminiferous tubules are creating fluids that fills the seminiferous tubule lumen and helping to push the spermatocytes along to the epididymis. The epididymis, these are structures that sit on top of the testes. And what they're doing is they're providing a storage area where these spermatids will sit until they are fully mature. All right. Once they're mature enough to leave, they exit through the vas deferens. That's going to transport sperm cells all the way out to the urethra. All right. And the urethra, remember when we talked about the urethra, not only is it a vessel that transports semen, but it also transports urine. We have 3 accessory glands that are producing secretions, are adding to the fluids. The first one is the seminal vesicle. This produces about 60% of the semen. The second one is the prostate gland, which produces about 30% of the semen. The seminal vesicles secrete what's called an alkaline fluid, along with fructose, which is sugar. The alkaline fluids, what they do is and the prostate gland is doing something very similar. There's also an alkaline secretion here. This is not only activating the sperm, but inside the vagina, that environment is very acidic, and it's so acidic that it can kill the sperm cells. So the alkaline secretions in the semen contributed from the seminal vesicles and the prostate gland both help to neutralize that acidic environment inside the vagina, and in turn helps the sperm cells to survive that environment. At the bulb of the penis, there is what's called the bulb urethral glands. And during sexual arousal, they'll produce a clear, slippery fluid that lubricates the inside of the urethra. So this is happening before the sperm cells arrive. And this has alkaline solutions in it also, but neutralizing the acidity that's from urine that still might be in the urethra. Here we see the prostate gland. And then up here we see the bulb urethral glands, 2 of them. The seminal vesicles are not in the picture. They'd be higher up. There's 2 divisions of the autonomic nervous system. So remember, the autonomic nervous system is that part of the nervous system that you really cannot control. We have the parasympathetic and then the sympathetic. For example, a good example would be that describes the functions of the parasympathetic and sympathetic nervous system would be if a bear were to walk in a room, that you were sitting in, a big grizzly bear, your body would definitely experience a lot of changes, and these changes would be outside of your control, right? So your heart would start racing, your breathing would increase, maybe your pupils would become dilated, your digestion would cease, you might lose control of your bladder, that kind of stuff. Again, you wouldn't have any control over this because it's the autonomic division of the nervous system that's causing all these changes. So that we would refer to these as the fight or flight responses, and they are controlled by the parasympathetic division of the nervous system, as is arousal. Sexual arousal is also controlled by this. However, if the bear were to get were to show that he's not interested in you and then leave, those changes, those physiological changes, are not permanent. All right. So everything would be restored. Now, getting everything to go back to normal, your heart rate has to go back to normal, your breathing rate, your pupils would go back to normal. These changes are governed by the sympathetic division of the nervous system. Okay. As is actually experiencing orgasm. All right. So during that time, the sperm cells will be propelled from the epididymis through the vas deferens, through the ejaculatory duct, down the urethra, and then until it is released, somewhere between 2.5 and 5 milliliters. And inside, there's somewhere between 50 million and 150 million sperm per milliliter. All right. So that's a lot of cells. All right. The female system, you can see the list of functions is a little bit longer. What is the job of the female reproductive system? 1, receive the sperm. 2, make eggs. 3, move the eggs from where they're made to where the sperm cells can fertilize them. 4, provide a site for development. 5, give birth. And then also there's hormones that are involved. So making the hormones that are associated with this reproductive process. So just like in males, the females have gonads, except the gonads now are going to be called ovaries. And the ovaries are going to be producing gametes. Not sperm cells, but egg cells. We call egg cells oocytes. Sometimes we call them ova. Eggs, all the same. That means all the same. The production of egg cells, we call that oogenesis. And there's hormones that are being produced by the ovaries. Estrogen is one of them. Progesterone is another one. There are accessory organs, uterine tubes, the uterus. There's some external accessory organs. Again, if we take a sagittal plane look through a piece of glass, we could see, let's make this a little bigger here. Here are the ovaries. So the egg cells are being produced there. From there, the egg cells would be released into the fallopian tube or the oviduct. The fertilized egg would then travel down into the uterus. Here's the muscle of the uterus wall. This is the thick muscular wall is called the myometrium. But the egg cell would find itself in the inside here. This is called the endometrium. There's a cervix here at the end of the uterus. And then we have the birth canal. The urethra is here, and it goes to the bladder. These are not reproductive structures. Again, same thing as before. This is mitosis. This is what's happening all over the body. This is for growth. Mitosis. Most of the cells in your body are produced by mitosis. Meiosis is how we produce sex cells. Why? Because we're taking diploid cells, this is a bad color choice, diploid cells, and producing haploid cells. And in this case, what's different with meiosis in females compared to males is males would produce 4 haploid cells. Females are producing 1 haploid egg cell from one parent cell. And the reason being is that that one cell needs to be really large. So it has to take the cytoplasm from these other ones. As a result, those other ones are going to be discarded. All right. So this process of oogenesis begins in utero. So this is happening as you are an embryo developing, the female is, as an embryo inside your mother's uterus. All right, so these cells become arrested. This meiosis process pauses after its first division and doesn't continue until puberty. All right, so these cells will remain in the diploid state until puberty. And then after puberty, they will each individually complete meiosis one by one, on a monthly basis to become haploid secondary oocytes. All right. So one haploid secondary oocyte is released from the ovary. We call that ovulation. And as it leaves the ovary, it sits in the fallopian tube. Fallopian tubes are also called oviducts. We might need to know that. Fallopian tube, oviducts. And then that cell will travel slowly towards the uterus. If the haploid egg cell that is sitting in the fallopian tube is fertilized by a sperm cell, then it will finish the process of meiosis too, and then it will become a zygote. So it's at that process of fertilization that the last division of meiosis happens, and then it discards that last polar body. The pronucleus from the sperm cell and the pronucleus from the egg cell will then combine to form a diploid zygote. Remember, zygotes are diploid because we have half the genetic material from the sperm cell combining with half the genetic material from the egg cell. So at birth, each ovary could contain somewhere between 200,000 to 2 million eggs. But do we release that many? Of course not. We're not alive for that long. If we only release 1 a month, we would never get to anywhere near that number. However, about 400 of these will actually mature to the point of ovulation. Again, oogenesis is produced as a result of hormones. Oogenesis is governed by the production of hormones. And again, the egg cells being produced and released happens as a result of negative feedback mechanisms. And the hormones are similar. It starts in the hypothalamus, releasing gonadotropin, releasing hormone, which is going to stimulate the anterior pituitary gland to release 2 hormones. The first one is follicle stimulating hormone. What this does is it takes immature follicles that are holding eggs, that are holding egg cells, and it stimulates them to mature. As these follicles that have egg cells on the inside mature, they become larger, right? This one's large. It's almost fully mature. It's when it looks like this here's the egg cell inside. Here it is. And you can see this follicle is filled with, this is called an antrum. It's filled with fluid. This is all fluid in here. When that antrum is big like that, that tells us this follicle is ready to release this egg. And when it does release the egg comes out into this area of which is the fallopian tube, and it's in that area that the sperm cells will be able to greet it and fertilize it, forming the diploid zygote. It's also at that point that the egg cell is going to be finishing meiosis with its last division of meiosis, and after that happens, it expels this polar body, which will degenerate quickly. What happens to the follicle after it releases the egg? What happens to these cells in here? Well, it becomes a structure. Now, this is a follicle, a mature follicle that has just lost its egg. It becomes this structure. This is called the corpus callosum. Corpus callosum. And it produces an important hormone called progesterone. We'll talk about that in a minute. Corpus, I said callosum, I meant corpus luteum. This becomes the corpus luteum, not the corpus callosum. Okay, the corpus luteum will produce one of the uterine hormones called progesterone. Another hormone coming from the uterus is estrogen. So there's two hormones that are coming from the uterus, two hormones that are coming from the brain, right? So these luteinizing hormone and follicle stimulating hormone come from the brain, from the pituitary gland. And they're playing an important part of what's called the ovarian cycle. So what does that mean? That means that they're taking these follicles, getting them to a point where they're mature, and then getting those eggs to be released. So it's these hormones that are responsible for that. Maturing the egg cells, maturing the follicles, releasing the egg cells. This happens as a result of follicle stimulating hormone, luteinizing hormone from the brain. All right. The other two hormones of the reproductive cycle are coming from the uterus in the form of estrogen and progesterone. Okay, but they're not having the effect of what's going, they're not having any influence or an effect on the maturing of egg cells and the release of egg cells. They're having an effect on structures that's somewhere else. So these hormones that are coming out of the ovary are actually having an effect on what's going on in here. And in here, this is the uterus. So these hormones are influencing a completely different cycle, not the ovarian cycle, but the uterine cycle, specifically when it comes to building up the walls of the uterus. The endometrial layer has to become thick. Every month it thickens and sheds. And that is controlled by these two hormones coming from the ovaries. We have hormones from the brain going to the ovaries, and they're controlling this. So we have two different cycles happening here. So follicle stimulating hormone, like I said, is released by the ant pit. All right. And this is stimulating the oocytes to mature into these large Graafian follicles. All right. That will then adhere to the edge of the ovary and allow that egg cell to be released into the fallopian tube. Luteinizing hormones are the hormones that are actually causing the Graafian follicle to ovulate, releasing that egg cell from the ovary. All right, so here we see the immature follicles developing. The follicle stimulating hormone is causing these follicles to grow and grow and grow until we have this large Graafian blister like, they call it blister like Graafian follicle that goes right up to the edge of the ovary, and then luteinizing hormone will spike, and that will cause the release of that egg cell. Corpus luteum is left behind. That's going to play an important part of maintaining the pregnancy. And if for some reason the egg cells do not become fertilized, then the corpus luteum will just degenerate and go away. All right, looking at this picture, this is a posterior view, so from the back side. So this would be the right uterus. I'm sorry, the right ovary. Here's the left ovary. Fallopian tubes have these little finger-like projections coming off of them. These are called the fimbriae. And what they do, as the egg cell is released, it's kind of floating out here, right? And these finger-like projections will kind of cause a current that will help pull the egg cell into the actual fallopian tube. So they'll be moving, coaxing the egg cell in. All right, so here we got the fallopian tube, and then this egg cell will begin to travel down the fallopian tubes until it ends up inside the uterus. The uterus has an inner layer here. This is called the endometrial layer. And this is where the egg cell will attach. There's also this thick muscle layer. This is called the myometrium. All right. And down here we have the cervix, which would be closed and until the baby is ready to be born, and then slowly it will open up. All right. So again, the fimbriae are important because the fallopian tubes are not connected to the ovaries. There's a space there. So those fimbriae will sweep that egg cell into the very beginning of the fallopian tube, so that it's in far enough that the sperm cells could actually reach it. After ovulation, it takes about a week for the egg cell to reach the uterus. And if that egg cell is not fertilized, the egg cell will just become broken down. All right, so the outer layer of the uterus is the perimetrium. Inside of that is that thick muscular layer called the myometrium. And then the internal layer, the site of attachment, is called the endometrium. It's that endometrial layer that is built up every month in anticipation of becoming pregnant, right? So it thickens and sheds every 28 days, and this is controlled by hormones, specifically estrogen and progesterone. So if there is no fertilization, then the endometrial layer is shed, and the result is menstruation. Sometimes fertilized eggs want to implant too far up near the fallopian tubes. There's not a lot of space up there to accommodate a large developing embryo and fetus. If that were to happen, the tube would start to stretch, and that can become dangerous for the mother because the tubes can rupture, causing bleeding. The embryo doesn't have a chance to survive if attachment happens up that high. So usually those pregnancies, unfortunately, have to be terminated. All right, so we have two separate cycles. Like I told you, the ovarian cycle starts with the release of gonadotropin releasing hormone in the hypothalamus, which causes the release of these two hormones. All right. So, what are these two hormones really controlling? Well, maturing of the eggs, maturing of the follicles, and ovulation. The uterine cycle is controlled by these two hormones. These hormones are called ovarian hormones because they're coming from the ovaries. And these are causing changes in the endometrium. So, getting the endometrial layer, the inner layer of the uterus, to thicken. Okay. With anticipation of a fertilized egg is going to be coming down and embedding itself. Again, follicle stimulating hormones stimulate the follicles to grow into those large Graafian follicles. Luteinizing hormones are responsible for ovulation. Estrogen, not only does it regulate development of secondary sex characteristics, but it also plays an important part in the uterine cycle, along with progesterone, also affecting development and influencing the thickening of the endometrial layer. Uterine cycle. Ovarian cycle. Controlling the events that are happening in the ovaries. These two control the events that are happening in the uterus. This is a pretty good picture. This is showing the events of the ovarian cycle, and this is showing the events of the uterine cycle. If we were to superimpose this on top, starting with luteinizing hormone, we could see a slow increase, and then it holds steady of follicle stimulating hormone. As it slowly increases and holds steady from day 0 to day 6, if we go back up here from day 0 to day 6, we could see these follicles getting bigger and bigger. Okay? And they'll continue to grow. Once we get to day 10, 12, it really increases here. Okay, now we're reaching about these follicles are going to start to look like ovarian, like very mature ovarian Graafian follicles. Look what happens also about day 11 and day 12. We see a spike in luteinizing hormone. So luteinizing hormone peaks at about day 13. If we go back up and look at day 13, what happens about the very next day? Well, it says right here, ovulation. So we see follicle stimulating hormone from day 1 to day 6, getting these follicles to mature. And then we see an increase in follicle stimulating hormone between days 10 and 12. And then we have a nice large Graafian follicle. Luteinizing hormone peaks about day 11. Day 13, it peaks actually, and then soon after we see ovulation. The egg cell is released. What happens to the mature follicle after the egg is released? It kind of lingers. It lingers behind and forms this corpus luteum. This corpus luteum is going to be producing progesterones and estrogens. All right, so let's look at estrogens. Estrogens are slowly increasing, steadily increasing from day 2, increasing. And then about day 12, it will start to decrease. Day 14, it's still relatively high, but on its way down. But what's happening is day 14, progesterone is starting to increase, right? Progesterone is starting to increase. So, okay. So let's look from day 2 to day 12 or 13. Day 2 is here. Day 13 is about here. What's happening here? Well, this is the prolific proliferative phase. So the endometrial layer is getting thicker and thicker and thicker. All right. As the estrogen levels come down, progesterone levels are increasing, and we enter into the secretory phase. So the thickness continues, and the thickness will be maintained as long as progesterone levels stay high. If this egg cell becomes fertilized, the corpus luteum will continue to produce progesterone, keeping this layer thick. However, if these egg cells do not become fertilized, the corpus luteum will degenerate and progesterone levels will stop. And that's going to trigger menstruation. So all this thick wall that we just spent the month building up using estrogen and progesterone will then be washed away. And that's going to take us into the beginning of our next period over here. We could see we're back to day 1 and 2, which is the same thing as what's happening over here. So we have the menstrual phase, followed by the pre-ovulatory phase. Then we have ovulation, and then we have post-ovulation, right? And we can see the estrogen is building up the endometrial layer during the proliferative phase. Progesterone is maintaining that layer during the secretory phase. And then, of course, is if there's no fertilization, then the corpus luteum degenerates, and then we're not able to maintain that layer anymore. It's going to be shed. So you got 4 hormones. These 2 controlling the uterine cycle. These 2 controlling the ovarian cycle. These are ovarian. These 2 controlling the uterine cycle. So getting the endometrial layer thick, right? Uterine. A little bit more advanced than the males. I really like this diagram. Along with this diagram, I think it's a great visual interpretation with a time scale. It shows the events inside the ovaries. It shows the events inside the uterus. It shows how each individual hormone is influencing the events that are happening in both of these places. I like it a lot. Birth control, there's only one true effective method of birth control, and that is abstinence. However, there are other birth control methods. There are surgical methods that use hormones, IUDs, spermicides, barriers, or just modifying behaviors. Each come with their advantages and disadvantages. When we're talking about surgical methods in males, that includes vasectomy. So you're taking the vas deferens and you are severing the vas deferens, preventing the sperm cells from getting from point A to point B, meaning that they are still being produced by the testes, but they are not able to exit through the urethra. They can't reach the urethra because the vas deferens has been cut. All right, so hormones are still being produced. Sexual desires does not change. It's just those cells are not being used, and they'll be absorbed by the body. In females, when we block the uterine tubes, that's called tubal ligation, and that will prevent the sperm cells from reaching the egg cell. So during ovulation, the egg cells will be sitting in here, but we've cut and tied these fallopian tubes, so as the sperm cells enter, they have no way of reaching. All right, same thing here. As the sperm cells are produced in the testes and stored in the epididymis, they'll try to leave, but they can't because we've cut this. All right, and then we have hormonal methods. I'm not going to read all of these. You can use the book and the notes to read about the different types of birth control, as well as the different types of STDs we see. I put a table in here that tells you the names of the STD, the scientific name, the common name, and then how they're classified, and usually they're classified on the type of pathogen that is causing them. Some symptoms and then treatments. You could see the ones that are bacterial related or protozoan related are treatable. However, the viral ones, not really curable. All right, so you can read that one as well. I'm going to stop this video here, and I'm going to begin chapter 6, what happens after fertilization, in a separate video.